Welcome to Blueprint of Life video 5. In this video, we're just going to quickly go over some of the secondary source or the investigation activities which you're going to be carrying out in class, but we'll just give a quick overview to give you some background before you complete those. So one of them is to look at technology and how it has played a role in evolution. So when Darwin first looked at natural selection, he used his evidence, or his evidence was based on comparative anatomy, embryology, and then behavior to support his ideas. We know that that wasn't the best method available, and as technology has improved, we've been able to change the classification of primates within the last century due to these advances. And biologists have now been able to analyze some evolutionary relationships and infer different ancestry, which we once thought and maybe which Darwin first proposed. And we're going to be looking at the three techniques which we discussed in video 4: amino acid sequencing, DNA hybridization, and DNA sequencing. The first is amino acid sequencing, and amino acids is a protein that is actually, when we use amino acid sequencing, we take out a protein which is found in a wide range of organisms and we study that one. The sequence of the amino acids in this protein is analyzed. Similarities in this amino acid sequence imply a shared common ancestor. Differences imply that the organisms have evolved, and the number of differences is somewhat proportional to the length of time since they separated. When we look at DNA hybridization, it involves applying heat to split double-stranded DNA molecules to expose the bases. We do that in the different organisms which we want to hybridize, and then we mix these separate segments. We allow the strands to cool and combine to form a hybrid. So we have one strand from one organism and one strand from another. And then we heat it again to test the strength and the relative combination of the hybrid DNA. So the higher the temperature, the more combined the DNA strands are, which indicates higher similarity. So here we have a diagram of how we can isolate from a human and a second chimpanzee. So we apply heat to form single strands. We mix these two single strands together, and then we heat it again to see how closely related they are. And we can see here that where they aren't bound means that they're not complementary base pairs, which means here is where these organisms have evolved and changed. And the third technique is DNA sequencing. So we take a piece of DNA, a specific gene usually, and it's isolated from each organism. And then we use some computer equipment called a DNA